right, folks, it looks like we've got a lovely small group here, which is fine, but I do want to, just Poreg reminded me that this is a pathology discussion. And so be warned that there will be some of the marine mammals showing the inside parts of them, not just the outside cuteness. I want to remind people that the Noya Center brings you these science talks as frequently as we can. We were able to bring some amazing speakers, which I feel just really above and beyond excited to have Poreg Duignan here from the Marine Mammal Center this evening. Someone who does the work that is very near and dear to my heart. I was just saying that that's really what got me into rescuing marine mammals, but I immediately was more fascinated about what their issues were. And so having the work that I have now in responding to the deceased one is about as close as I can get to working in a necropsy lab. And so I often clarify, I don't do a necropsy in the field. I simply don't do that. That's something that the professionals do, that someone like Poreg will do. I will do a field necropsy and measure the animal and look for obvious things, but the deeper work is done by people like Poreg and team. So if you are able, please do drop a donation into our, Trey will put the link up. We do ask for a $10 donation for our science talks. Just another great way to support the Noya Center. And for those of you who are already members, we really want to thank you. It keeps us chugging along. And for those of you who are not members yet, think about joining and becoming a monthly sustaining member. And that way you'll get our newsletter, our Pinniped Press. Many of these, we're growing in our story, in our written content, and sharing out some really good science. So yet another reason to become a member. You also can go free into the Discovery Center and get a discount, I believe. With that, I hand it over to Poreg, and thank you again for being here.
Thank you, Sarah, for the invitation, and thank you for being such a great collaborator. So I'm just going to call this up. Now, hopefully you can see that.
Perfect.
Okay, great. So what I'm going to talk about is some of the diseases that we see on the West Coast here, and try and pick out things where, you know, they're either natural events or whether the climate is involved in the occurrence of these diseases or the severity of them, and what environmental and human activities might give rise to them. So all the kind of disease ecology is what is of interest to us, what's happening at the population level, not just at the individual animal level. So come on, move. Okay, so what I thought I'd start with is just give you a little bit of background about me, where I've come from, how I got to be the director of pathology at the Marine Mammal Center, which is a fairly unique position and something I'm very privileged to have. So you probably guessed by now I'm Irish. So I was born in Ireland. I did all my primary degrees in Dublin. I have a BSc degree in zoology. I did a master's degree in biochemistry, and then I did follow that with my veterinary degree. So a lot of time in college in Ireland. Then straight out of college, I went to work in Saudi Arabia. I worked in avian pathology, did some wildlife work there. And that was really just sort of a little hiatus for a year because I'd already planned to go to Canada to do marine mammal pathology because that had always been my dream. And for that, I went to the Ontario Veterinary College because at that time in the 1990s, it had not only an excellent pathology school, but it also had the only professor in a North American university who actually specialized in marine mammal health. So he became my PhD advisor, and I did pathology residency and PhD and spent 6 years at that lab in Canada. After that, I did another year's pathology residency down at UC Davis, and that was to just broaden my field of expertise in different species and different climatic zone and everything. And from there, I got picked up for a faculty position at Massey University in New Zealand. And that was actually one where they were looking for somebody specifically to do marine mammal stranding research, which was pretty unique. So I jumped at that position. And then while I was there, I spent 9 years in New Zealand. And during that time, I founded a New Zealand Wildlife Health Center based at that university, and we did not only marine mammal work, but all native species like native endangered birds and other things too. After that, I returned to Ireland for a couple of years, worked for the Department of Agriculture and Foreign Animal Diseases, because my specialty in my PhD had been viral infections, so I did that for a couple of years. Then did some diagnostic pathology work at a lab in the UK where, again, they did a lot of wildlife, and got tired of working for government. So I went back to teaching faculty work at the University of Melbourne in Australia. And I did all the wildlife surveillance work for the Victorian Wildlife Health Network and continued marine mammal research there with a bunch of different collaborators I had known from when I was in New Zealand. Then I returned to Canada because they opened up a new vet school in Calgary, and they were looking for a wildlife pathologist to start a new program there. So it was a very good offer, brand new vet school, well funded, and gave me the opportunity to work back in the Arctic again with Arctic marine mammals. And from there, I was recruited by the Marine Mammal Center 7 years ago now. So I've been director of pathology at TMMC since June 2016. And I have a small but very good team. So now we have 2 pathologists. So after I started at TMMC and realized, what a huge caseload we have there and what interesting cases we get, I created a pathology fellowship, and Maggie Martinez, who's on the right of the picture there, she was the first fellow I hired, and it was meant to be a 1-year position, but Maggie already came with a completed pathology residency, so she had her pathology boards completed, and she had also completed a PhD, so I couldn't ask for any more. That's about as highly qualified as you can get for an early career pathologist. So we kept Maggie, and she is now our staff pathologist. So we have 2 pathologists, 2 necropsy technicians, Jackie and Barbie, and 2 lab technologists. So Carlos, who's the lab director, and Jennifer, who's the second lab technologist. Jessie Rose was in that position, but she's moved to another lab now. So small team, but very effective. And you all know where we're located. We're down in Sausalito, and we have a stranding range that covers about 1,000 kilometers or 600 miles of the California coast from where you guys are down to Morro Bay. And alongside our main hospital in Sausalito, we have 2 satellite hospitals, one at Moss Landing in Monterey Bay and one at San Luis Obispo or Morro Bay. We also have a hospital in Hawaii, but I'm not going to talk about that one because we don't really do that much pathology for them. So why study marine mammal pathology? People always ask me. Well, there's a lot of similarities between us and them. We're both mammals. We get similar diseases. We're exposed to similar toxins and respond in similar ways to them. We also are basically top predators. We eat the same kind of food. Sea lions, for example, eat a very similar seafood diet to what we do. Everything from fin fish to octopus and squid, the whole range of things. The only thing they don't eat is maybe shellfish, but otherwise very similar. Socially, marine mammals are quite similar to us. They're quite social animals. Disease epidemiology or the spread of infectious diseases can be very similar between them as between us. And we are exposed to similar new and emerging diseases that marine mammals are, like coronaviruses. I thought the one that's caused the recent pandemic in humans. Similar viruses occur in marine mammals. And climate change is impacting them as much as it is us. So there are lots of commonalities there and lots of good reasons to know more about what's happening in the ocean. And some of our research covers some of these really important diseases. And Again, similar to what people get. So cancer research is a big thing for us. Feline urogenital carcinoma is one of our top diseases, top focuses of research. And then just very recently, we discovered a whole new type of cancer in northern elephant seal pups. It's a type of leukemia lymphoma, again, with similarities to neonatal or infantile leukemia lymphoma in children. Evolving and emerging infectious diseases are a big thing for us. And of those, there are bacterial diseases, viral diseases, protozoal diseases, and parasites. And of the bacteria, Leptospirosis in sea lions is probably one of the biggest ones. You've probably heard about that. And it's very similar to Leptospirosis in dogs, which you're familiar with if you have a dog because you vaccinate your dog against it. But people can also get it, and in people it's called Weil's disease. Klebsiella pneumonia, also a human disease. It started as really a major human type of pneumonia, and up until the advent of antibiotics was the #1 killer of people when they got pneumonia, was this particular bacterium. Now we have a strain of it particularly in our sea lions. That's one of the most significant diseases we're dealing with right now. Pasteurella is another bacterium. It's commonly called avian cholera because it's spread by waterfowl like ducks and geese. And you get big outbreaks in their populations when they're overwintering. But marine mammals also get exposed to it. Brucellosis coccidioides is a fungal infection. It's valley fever. So anybody who's familiar with the Central Valley and the southern part of the coast, that's where that occurs. So it's a systemic fungal infection, like a yeast type infection. Then I mentioned the coronaviruses. So we're familiar with our recent pandemic virus, but marine mammals also have a number of coronaviruses of their own. Morbilliviruses are huge in marine mammals, not so much on the West Coast. So I'm not going to talk about them here, but in the North Atlantic, it's caused major epidemics in both seals and dolphins, also in the Mediterranean, Australia, more recently around Hawaii. So it's a cause of mortality for pinnipeds like seals, but also for dolphins and even the whales. And as I said, we don't see it so much on the West Coast, but it is here, but we just don't see big outbreaks of it. And then the protozoa are a big thing on the West Coast. So Toxoplasma, the disease that is acquired from cats, and Sarcocystis, which is another protozoan, a cousin of Toxoplasma, also acquired from a terrestrial animal, but in that case, the possum. And then some parasites can be new and emerging problems. And one in particular is this hepatic capillariasis that we have recently identified as a cause of mortality in sea otters, and otostrongyliosis, which is a nematode, lungworm, and vascular parasite of northern elephant seals. Toxins, there's a number of really important harmful algal bloom toxins. Domoic acid is the big one on the West Coast, but saxitoxin is also here and also on the East Coast. And this new one, fluorosis in sea lions. And now this is one where a case that Sarah alerted us to, and then some subsequent cases, is from your part of the woods. This is a Mendocino disease, so we'll talk about that. Influence of environmental change on health. We have a number of different projects. We're looking specifically at how climate and environment change is impacting on the health of particular animals in particular places. So one we're looking at is the narwhal in the Arctic, because that's a place of rapid climate change, and the other is in Gulf of Mexico, where changes in the water quality, particularly the freshness of the water, is affecting skin in coastal dolphins and causing mortality in them. And then there are unusual mortality events. So again, sticking to the West Coast, the gray whales obviously is the big one here, but we've also had Guadalupe fur seal UMEs, and then more north of us, Arctic foetid alopecia syndrome. But anything there with a red tick is what I'm going to talk about in a little bit more detail. Okay, so starting with cancer, the main one that we see is C-line urogenital carcinoma, and we just abbreviate that UGC. And it's the most similar to human cervical cancer that you could possibly imagine. It's almost identical to that in how it presents. So what's new about it? What have we found? Where are we going with this research? And how, how is it related to cancer in people? Those are all questions that we're trying to work on. And then other kinds of cancer, well, marine mammals get various kinds of cancer, just like people do. Like any particular organ can develop cancer. I mean, that's what happens. But the prevalence of those or incidence of those cancers is very, very, very low. This leukemia lymphoma in northern elephant seal pups is something we've just discovered in the last 2 years. So it's a completely new cancer. And again, it's virus associated, kind of like the urogenital carcinoma is in the sea lions. Okay, so UGC is a multifactorial cancer, like a lot of cancers are. So there's something that will induce the cancer. In this case, it's a herpes virus, so you need to have that. And the herpes virus, we now know from our research, carries cancer-causing genes or oncogenes. So that's what you need to get it started. But then you need also something to promote it and actually, really get it going. And that promoters are something else, like pollutants or chemicals or, like smoking in people, for example. But in this case, persistent organic pollutants is what are the promoters for marine mammals. So those are like DDT, PCBs, all those kind of legacy type chemicals that are man-made and don't break down very well, but cause adverse effects in whatever system they get into. And then genetics may play a role. Our recent big 20-year study has sort of downplayed the role of genetics a little bit from some earlier work we did. But the two big things are the top two there, the herpes virus and the pollutants. And we see an awful lot of this cancer. So of all the sea lions that we admit to our hospital at Sausalito and die, so whatever reason they die for, whether it's trauma, whether it's domoic acid poisoning, leptospirosis, or something else, of all of the adult sea lions that die in our necropsy, 25% of them have this cancer, and that is a huge amount of cancer. And about 18% of those will have metastatic disease that directly causes their death. Either they come in so sick they have to be euthanized because of it, or it directly kills them. And then the remainder that makes it up to the 25%, these are animals that die for some other reason, like they have a shark bite or something. But during the necropsy or the histology, we will find the cancer in them as well at an earlier stage that later on presumably would go on to kill them. But at that point, it's an early stage cancer. And to put that all in context, I mentioned that this cancer is very, very similar to cervical cancer in people. And if you look at, how that's rated in terms of severity, it's the number 4 killer of incidence and prevalence of cancer in women. And the case fatality rate is regarded as being very high at 13.3 deaths per 100,000 people globally every year. So that would be an incidence rate of about 0.013% compared to the 25% we're seeing in sea lions. So that just puts it in context of how enormous this is for sea lions. So this is a figure from an old paper of ours going back a few years now, but it just shows you that year on year, the prevalence of cancer we see in our population varies a lot. You see some there as low as 11%, in some years it's as high as 51%, but that all depends on the intake of sea lions to our hospital and what they've come in for, what else is happening. So whether there's algal blooms, whether there's El Nino events or other things happening. So that just reflects that. But basically that 25% I mentioned is something that's been pretty consistent over the last 30 years. That has not changed substantially. This year, we've actually seen an awful lot of cancer, and particularly in June and July of this year. So that pink line there is this year. So you can see every other year, well, for the last 7 years, it's sort of tracked along like this, sort of very steady intake of cases as you go through the year, and not too much variation from year to year. But this year in June, July, it just seems to have shot up here, and we already have passed the 20 cases by the start of July. So who knows where it's going to end up. But I can't explain that yet. We'll have to do pathology on all these animals to see what else is going on. Did they get admitted because they had domoic acid poisoning or leptospirosis or something else? And that's why we got them in and then discovered the cancer, because the cancer wasn't the primary cause for them dying in each case. But I have to do all of that analysis yet. Just interesting that we've had so many in the last 2 months. Okay, so just give you some examples of what the cancer looks like, how these animals present. So often they'll be like, this is an adult female. You can see she's in kind of slim body condition, and that's often a feature of cancer, whether it's this cancer or anything else. It's called cachexia of cancer. Animals with cancer don't, people even lose appetite. They don't eat so much, or they don't metabolize their food so well, so they end up losing weight. And sea lions are exactly the same. And they'll often have edema or buildup of fluid in the back end because of the cancer blocking off lymphatics and causing the buildup of fluid. So here you can see this swollen vulva is the manifestation of that. Males, the same thing. This guy, again, you can see he's very poor body condition, and he's got the swollen hind end. This time it's the scrotum is all swollen with fluid in the hind flippers. Now, this is where it gets a little bit more graphic, so I hope that doesn't bother you, but this is the urogenital tract of an adult female sea lion. So the whole, everything from the kidneys down to the vulva. So in the middle there, you've got the 2 kidneys, above it, the adrenals. So this is all taken out en bloc. So everything is still attached. So you've got the sublumbar lymph nodes here. Those are the 2 ovaries there and there. These are the horns of the uterus, the body of the uterus, the cervix, and the vagina. And then off to the side here is the urinary bladder. And if you look a little bit closer at this area, the vagina and the cervix, that's what's over here on the left. This knobbly thing is the cervix, and this is actually, you just have to take it from me, it's too knobbly. It's got all these little bumps on it. These are all cancer nodules, and some of that is here in this proximal part of the vagina too, this thickened lining. This is all because of cancer. And then this is where it starts. So it starts either in the cervix or the proximal part of the vagina in a female. It's the penis or the prepuce in a male. And from there, it metastasizes or spreads very rapidly, either through the lymphatic system or through the blood vessels to other organs. And if it goes in the lymphatics, they drain to the lymph nodes, which are here, the sublumbar lymph nodes. Now, you should not be able to see these like that. These are really enlarged. And a consequence of them getting so enlarged is that they will block off the ureters, draining the kidney down to the bladder. And that then forces urine to back up into the kidney, turning the kidney into this Swiss cheese hydronephrosis. And often these sea lions will present clinically as renal failure because the kidneys are failing. Now, the metastases can also go to the liver. Here's multiple slices of the liver. All of that pale stuff is the cancer in the liver. Here it is on the surface of the kidney. That's a section through those lymph nodes. It can also go to the lungs, and it can also go into the spine and into the pelvis, and they can get pathologic fractures of the spine and pelvis, which is not a very pleasant thing. In the male, here's again the urogenital tract taken out completely. So you've got the 2 kidneys with hydronephrosis because of the large lymph nodes blocking off the ureters. And in this case, the primary is going to be somewhere here in the prepuce or on the penis, and it can be really difficult to find in a male. And here's this little tiny little nubbin of a nodule here. That's the primary site in a male. But from there, it'll metastasize rapidly and widely in the body. So what do we see when we look down the microscope at this? Well, the changes in the lining or the epithelium of those organs is quite apparent. So this is in a cervix, and you get a transition here from kind of normal epithelium over here on the right through dysplasia, which is just disorganized jumble cells forming that epithelium, into frank neoplasia, which is this big nodule here. So this is still within the epithelium. So we call this carcinoma in situ. So it's in the epithelium, it's still contained, it hasn't invaded beyond that or hasn't metastasized. But very soon after that, it will invade. And here we have invasion. You can actually see these are lymphatics, these little clear spaces. These are lymphatic vessels, all plugged with cancer metastases. So these little nodules of cancer cells are now spreading through the lymphatics to get to other organs. And here's a close-up of one of those. And you can see how crazy these cells are. They're all different sizes and shapes, some with very weird cells and huge nuclei or nucleoli, very, very irregular looking. So those are the hallmarks of a malignant cancer. Now, so what are we doing about research? So the big questions we had were what factors are involved? Like what were the primary causes of this, and what other factors are involved in it? So the basic epidemiology of the cancer, and then what are the mechanisms? So if we suspect the virus is involved, how is it doing that? What are the features of that virus that allow it to do that? So it's actually this cancer was first discovered in the late 1990s, and it was very soon after that the herpes virus was identified and the high levels of pollutants were identified, but the direct association with the cancer had not been made. It took years and years to actually make those associations. So one of those studies was this PhD study by Dr. Alyssa Deming, who is now she now works as a veterinarian for Pacific Marine Mammal Center down in Southern California. But she was a research fellow at the Marine Mammal Center when she did this PhD through the University of Florida. And then a second key study was this 20-year case control study that was led by TMNC, but involved a whole bunch of different collaborators and different institutions with hundreds of animals, both cases and controls, to look at the factors that contributed to cancer in those. Okay, so from Alyssa's study, part of what she did was another like a 10-year study on the incidence of cancer. And then between her work and two previous studies, we know that this prevalence pretty much has held up over the years, that roughly 25% of our cases will have cancer if you factor in all the animals with just early and histologic lesions. So that's something that hasn't changed very much. The main thing that Alyssa was able to do, though, was to look at the role of the herpes virus in these. So it's a garment herpes virus. It's called OTHV1. That's otorine herpes virus 1. And a neat thing about it that she found when she sequenced it was that it has very similar cancer-causing genes or oncogenes to Kaposi's, the virus that causes Kaposi sarcoma in people, and that's human herpes virus 8. So they both have about 3 of these oncogenes, and she was able to find these in the sea lion virus, which is very unusual because of every species has got herpes viruses, and some have got multiple, like humans have at least 8. And most animal species have, but very few have cancer-causing herpes viruses that have these oncogenes. So this is a rare thing. And the cervical cancer I mentioned in people is also virus associated, and you probably are well aware now that there's a vaccine out against human papillomavirus to prevent that in people. And that has actually made a huge difference in the incidence of cervical cancer in humans, because in developing countries where that vaccine is not as widely available, the prevalence is still really high. But in developed Western countries, it's dropped enormously because of this vaccine. So what Alyssa was able to do was she took cancer cases that we had at the center and looked at healthy controls, animals that had CIN or carcinoma in situ, that early stage where it's still in the epithelium, and deeply invasive carcinoma that's already started to become malignant. And what she looked at was the expression of messenger RNA from these three different oncogenes. So that the expression of messenger RNA shows that these genes are active. They're actually producing a product that you can measure or detect in the cells. So she was able to show that messenger RNA from these three different oncogenes is present both in the carcinoma in situ and in the deeply invasive carcinoma, but it's not present in the healthy controls. So that is about as close as you can possibly get to fulfilling Koch's postulates for a cancer as you can get, because clearly we can't create laboratory sea lions that are free of everything and then stick this virus into them and see if they get cancer. That's just something that could never happen ethically or morally. So this is as close as you can get to saying, yes, this virus is implicated in this cancer. Now, the other study that I said was a key recent finding, and this paper was published in 2020, and this was the large case control, so basically a 20-year case control study looking at all the different possible factors that could be present in the animals with cancer. So including the virus, presence of the virus, levels of toxins, their genetic status, and age, and all these other factors. So this multi-year case control, then hundreds of animals involved, and it did confirm herpes virus infection was one of the key things, and also the levels of pollutants that they acquire from fish. So 394 sea lions were included in this. About half were cancer cases and the other half were cancer-free. And all that was based on necropsy and histology. So they all had both to rule out or rule in the presence of cancer. And then all these different factors were looked at. So over that study then, for every unit increase in the level of these POPs, or persistent organic pollutants, present in our body fat, the odds of developing cancer increased by 28%. And if an animal had the virus plus a low level of POPs, then there was a 30% chance that it would get cancer. But if it had the virus plus a high level of POPs, then it was more than less likely 80% likely to get cancer. So clearly you needed both. You needed the virus to trigger the cancer and then the POPs to actually promote it and turn it into a clinical syndrome. An interesting thing is that California sea lions also occur in Mexico. There's a population down in Baja, Mexico. They're a little bit distinct from our population here in California, so they don't mix very much. But in Mexico, they have the same virus. They have the OTHV1 virus, but they never see cancer. And that the difference there is that they don't have the pollutants that come from Southern California, where our sea lions breed. Okay, so herpes viruses and DDT, those are the big things. And you may remember 2 years ago or so, there was the big expose done by the Los Angeles Times based on research by David Valentine's group at University of Santa Barbara, where Valentine and his colleagues used submersibles, these unmanned submarines, to go look for where DDT had been dumped in the California Bight between the 1940s and the 1970s. So they did that, and they found these dump sites documenting thousands of barrels of dumped waste down there. And then Rosanna Shaw wrote this article in the LA Times, and it sort of all blew up in the media, this big storm about it. And it was actually right after our 20-year study had been published. So this is January 2021. that paper I just talked about was published in December 2020. So it all kind of coincided with that. So what we know about these DDT hex chemicals, like there's more than DDT, there's like 45 or so different chemicals in this mix, and they just call them out DDT X. So they're already known to have reproductive effects, and that's because they're endocrine disrupting chemicals. So that was already known from previous studies. They're also immune modulating, so they affect the immune system directly by affecting lymphocytes in the blood, and they can therefore affect the prevalence of or severity of disease because they're affecting the immune response. They can also, it's also suspected that they enhance seizures in animals with domoic acid poisoning. And then obviously this link with cancer, not just in sea lions, but also suspected in humans as well. So based on what happened or transpired from all of that exposure about the dump sites, UC Santa Barbara, where David Valentine is, they hosted this workshop in May of last year called the California Coastal Chlorocontamination, or C5 workshop. And lots of people were invited to that, including us because of our sea lion work, but also everybody, like geologists, water chemists, a whole range of people, and also people representing human health, social sciences, indigenous people, you name it. It was a very diverse group of people. And so the first was a workshop in person at Santa Barbara, and then there were 2 follow-up Zoom meeting type workshops. And the outcome of that actually, and government was represented at that, the California government and federal government. And the outcome of this actually was that the California state government has put $6 million up for research. And a bunch of us got together, and this is how much DDT is being dumped down there. So 1 million gallons of DDT waste generated by the Montrose Chemical Corporation every year, and all that dumped into the ocean over that period of 30 to 40 years. So it's a heck of a lot of waste. And this is where they dumped it. So the Montrose plant was right here on this Palos Verdes peninsula. They dumped waste directly into the sewage system right onto the Palos Verdes shelf, which they weren't supposed to do, but they did. And then where they were meant to dump the waste was by taking it out by ship to the deepest part of the canyon here between the Channel Islands and the coast, and dumping it there in deep water, like 1,000 feet plus of water. And 2 of the dump sites here have a lot of these waste barrels, but they also dumped barrels on the way to that site. They didn't always go off the shelf. They just barely got off the shelf and dumped the barrels all the way between the 2. So there's waste all over this region. And the effects of that are now pretty well documented. The highest concentrations of DDT in dolphins has been documented here. They also, the group at San Diego State, that's Wenaho professor down there, and Nathan Dollar, both at San Diego State University Toxicology Lab. And they also measured levels of DDT in condors, so part of the Condor reintroduction program. Now, historically, condors, as you know, would have preyed or fed on marine mammal carcasses along the California coast. Sad thing now is that those carcasses are so, particularly sea lions, are so loaded with DDT that it's having the same effect on their reproduction and eggs as it had on peregrine falcons back in the 1970s, when it almost wiped out peregrine falcons. Same thing's now been discovered with the coastal condors, is that their eggshells are too thin to be incubated, and those nests are failing. So a really sad consequence, for a species like that that's just come back from the brink. Anyway, so the outcome of those workshops last year was that California State put $6 million into a fund that's managed by Sea Grant, California Sea Grant, and then that was open for bids for research, and we were one of the parties along with Ho's lab at San Diego State University and some other people to get some of this money. But other projects that we're also trying to develop is developing a pap smear type test for sea lions to detect early cancer in them. We're also looking at further into the genetics of the animals and the virus with a collaborator in North Carolina State. And we're looking for collaborators to actually study the mechanisms of carcinoma metastasis, because this is not well known either for anything, not just sea lions, but even for humans. How does metastasis happen? How do cells break away from the primary tumor, get into the circulation, and then set up home in a different organ? How does that work? How does that happen? Because that's fundamental to how these cancers actually kill animals or people. It's not the primary cancer that kills them, it's where the metastases go. And then this last one here, the role of the newly discovered DDT related compounds, and there's about 45 of them in causing adverse health effects. So we put together a consortium of people, including that lab in San Diego State. And so we're looking at marine mammals, condors, laboratory models for the effects of these chemicals using zebrafish, but also looking at health risks in people. So all the sort of multifactorial risk analysis kind of research. And that project has just got funded, and we've only just started work on that now, and that will be carried out over the next 18 months. And hopefully it will, the results of that will then lead to further research and hopefully more funding from both state and federal agencies to try and get to the bottom of this. Now, I mentioned that we discovered a new cancer, and this one is also virus associated. It's also a gamma herpes virus that we just discovered in northern elephant seal pups, and we had 2 of these guys in 2 different years, one from Santa Cruz County and one from San Luis Obispo County. Now, these guys presented with first of all, it was diagnosed as leukemia, so excess white blood cells circulating in the blood. And then when we suspected those animals had cancer and they were euthanized, we confirmed that they had lymphoma, or cancer spread throughout the lymphatic system in the body. So that was published, and this is just a figure from that paper. So these are the 2 flippers of a northern elephant seal pup, one of those 2 that we had. Huge lymph nodes here with sort of a homogeneous appearance that you get with lymphoma. So it's completely taken over by this population of neoplastic or cancerous white cells. Lymph nodes throughout the body and the spleen. So this is this huge spleen, and these are all the lymph nodes here, all expanded by these neoplastic cells. And then when they're stained to show what kind of cell they were found to be B cells, so a uniform population of neoplastic B cells. So we found with a collab, we use a virology lab in University of Florida as our virology collaborators. We sent tissue off to them. They found this brand new gamma herpes virus, which they've called mirror gamma herpes virus 3. So Mirawanga is the genus name for the elephant seal. So Mirawanga gamma herpes virus 3. And the question now is, does it also carry the same oncogenes like the sea lion virus? We don't know that just yet. And what role did the contaminants play in this, like it does in the sea lions? Again, we haven't got the answers for that yet. And how prevalent is the virus in the sea lion population? Well, we've made a little bit of progress on that because we have developed this new lab assay here based on the sequence from that virus. So we can now use this PCR test with blood samples or tissue samples from northern elephant seals to see whether or not they have the virus, and therefore get an idea of how prevalent it is in the population. Okay, so infectious diseases, our leptospirosis. Lepto has been around in sea lions forever. It was first diagnosed back in the 1970s. And for many, many years, it just seemed to cycle every 3 or 4 years. There was an outbreak, and then it would go away, and then would be another outbreak. It would never completely go away in between. There was always some cases. But a weird thing happened in the last decade, and that was this fade out phenomenon. And it's something that nobody had predicted. So we had a big outbreak in 2011, and then nothing until 2017. And during that interim, when it wasn't causing disease, it was completely gone. Like all the surveillance work to look for it in the population, to look at antibody levels in animals, showed that it was just completely gone. So there was something that nobody had ever thought could happen. So now we know that's probably linked to what's happened in the North Pacific over that period of time. So those blob events, those thermal anomalies that have happened, probably had a big role in that. And the funny thing is too, when it did reappear in 2017, the pathology we're seeing seemed to be different from before the fade out. Okay, so what does it look like? So when we get a lepto animal, first thing we'll do is a blood test on it and see what the kidney values are. So the chemistry of the blood as affected by the kidney. So blood urea nitrogen and creatinine and phosphorus, all of those things will be really high, showing renal failure. So that's an immediate indicator that the animal is leptospirosis. So if it dies then, and we do necropsy on it, we can clearly tell the difference between a lepto kidney and a normal kidney. So normal kidney is like this one on the left, and a normal kidney anatomy for marine mammal it's a reniculate structure. It looks like a bunch of grapes. So all those little individual units is each its own little kidney. And it has its own cortex, its own medulla, and its own pelvis. And then there's hundreds of them in each kidney. And when you get leptospirosis, what you see is expansion of the cortex. So this pale tan should be darker, and congestion or hemorrhage at the corticomedullary junction, and then down to the papilla, which is normal. But this distinction between an expanded pale cortex and the bright red corticomedullary hemorrhage or congestion is what really shows us this lepto, or high suspicion anyway. Histologically then, what you're seeing in the cortex is necrosis or death of the tubule cells. So the glomerulus is here, and then the urine's filtered out from there into this space, and then it goes through these tubules, and the water and some of the good stuff is reabsorbed and the bad stuff's excreted. But if these tubules are knocked out like this, none of that can happen. So the bad stuff all stays in the blood. So this tubule here is completely gone. These ones are affected, but less so. And then there's lots of inflammation. All of these cells here are white blood cells. So that's called a chronic interstitial nephritis with tubular necrosis. And we can tell it's leptospirosis if we do special staining for it. So this is like a silver stain. This little tangle of black stuff here is leptospires. Or if we do a very specialized immunohistochemical stain that specifically is for leptospiral antigen, then all the red here, this is all Leptospira pomona, the cause of leptospirosis. And it's all associated with the tubules. It's not in the glomeruli. The glomeruli here are completely normal and not affected. Okay, so all of that is stuff that we have known from years ago, that you get the tubular interstitial nephritis, you can demonstrate the leptospires are there in the tubules, and you get this mixed leukocytic inflammation. So that's all stuff that was in the literature from a long time. What we saw starting in 2017 was this uremic syndrome, where you get this plus this. So this is all the other damage caused to other organs by the uremic toxins circulating in the blood. So you can get ulcers in various places like the mouth, respiratory tract, urogenital tract. You can get interstitial pneumonia, and you can get dermatitis and even brain edema. And that manifests like this. So you get these buccal ulcers on the gums and the lips. You get bleeding gastric ulcers, so these big holes are bleeding ulcers. The brown is blood that's just been digested by the acid in the stomach. Here's a huge blood clot in the stomach where it's bled out from one of those ulcers. And then you get this sloughing skin. This is on the flippers, and this skin just deglobs off. I mean, you just touch it and the skin falls off. The lungs can get this severe interstitial edema. So all of this gray, hazy stuff in between these little lobules is fluid leaking out of the blood vessels in the lung. And then that sets up a perfect environment for bacteria to cause bronchopneumonia. Okay, and then so the fade out and the reemergence. So I said that it used to occur at a frequency of like 3 to 5 years regularly. And then in between, you just get some individual cases, but you wouldn't get big outbreaks. And then with this fade out that happened after 2013, or between 2013 and 17, it just disappeared from the population completely. There was no antibodies in the animals at all. Pups that were born in that period did not get infected, did not seroconvert. And then there was a little outbreak in 2017, and then a huge one in 2018. So there you can see that regular pattern of outbreaks, you know, from the early 1980s right through to 2011. And then the fade out happened over this period of time, and then it reemerged. And now we had another big outbreak back this past fall, and that's continued right through this summer. So the question was, and luckily there was a group at UCLA who are doing the epidemiology of leptospirosis as their main research. They started a big NSF grant around this time, 2012. And initially they were in a panic because it just disappeared and they had nothing to study. But what they did do was actually study this fade out phenomenon and get to the bottom of what was going on. So their research has shown pretty clearly that it's very closely linked with these blob events, these thermal anomalies that happened in the North Pacific, and there were 2 of them. One started about 2013 and went through 16, and then another one started 17 or so, went through 19. So both of them messed up the ecology of the North Pacific in a big way. So 2 possible ways that it did was, one is that it drastically affected pup production down in the Channel Islands, and particularly in 2015, the pup production practically crashed. There was no pups came off those islands at all in 2015, or the pups that did died very soon because they had not been fed by their mothers, and they never made it up the coast. So they didn't maintain the cycle of transmission between older animals and younger animals. And then another thing that happened was, or presumed to have happened, is that the prey distribution, because of the change in water temperature varied. And you mentioned earlier how the whales in 2015 were numerous because of the blob and all the anchovies coming inshore. Well, all of that changed the way sea lions were feeding and mixing, and all of that changed to the dynamics of how leptospirosis was spreading in the population. So there's those 2 big marine heat waves. So this was the one in 2014, well, it started in 2013, went through to 2016. This was it in September 2014. So all of the red and orange there is temperatures above normal. And then again, 2019, again, all temperatures above normal. And then something new from last year was that we've now found leptospirosis as a field acquired or naturally acquired disease in northern elephant seals. We did know from previous work that if they got exposed to infected sea lions, like in the hospital, then they could get it. But we didn't know they could actually get it as a natural infection in the wild. And from the last year, we now know from both Washington, Oregon, and California cases that that can happen. They can actually be picked up from the beach with active leptospirosis infections. So we've documented a bunch of those, and it's exactly the same as leptospirosis in sea lions and the same strain of leptospira that sea lions have. Another bacterial disease is Klebsiella pneumonia. Now, this one is quite interesting. I first came across this in New Zealand in 2002, and that's when I was working there at Massey University, and I was leading up this sea lion research program in the sub-Antarctic. And fortunately, we had started this health monitoring program in 1998, so 3 years or so before this bacterium ever showed up. And when it did show up and caused epidemics, we were right there and on top of it. So these are some graphs of pup mortality from the Auckland Islands seal colony at Enderby Island. And you can see this dark line, it was the pup mortality curve for 2001. It's like down here. And then 2002 and 2003, it's way up here. And like 50% of the pups died in those 2 years. 2004, it had beginning to abate a little bit. So what we found, and we had archived tissues and materials from the previous year, so we knew from testing those that this organism, when we isolated it, was something we'd never seen before. And the presentation was something we had never seen before. So what these little pups had was this massive bacterial suppurative infection. You can see very swollen flippers here. When these little guys died, it's just all pus and all the tissue. You see, this is the actual radius here. The bones are just falling out of the flesh. Really, really nasty. All the muscle here involved, the joints. You can see here again, necrotic bone. This is the radius, just necrotic pus pouring out of these abscesses. So very nasty. Also affected the skin because there's like flesh-eating skin disease and also caused meningitis. So at the time, I had just taken on this new PhD student, Aurélie Castanal from France. So immediately put her on to working on this as a part of her PhD project. And we were able to do a lot of work on it. But subsequently found out that around the same time as we were having this, so early 2000s, an outbreak of disease had occurred in California sea lions down at the Channel Islands and San Miguel, but they hadn't done the earlier work with health down there to know whether this was new or not, and you know, so the paper they wrote on it back then more or less implicated hookworms as the cause, with the Klebsiella as being a secondary infection. But you know, we now know that isn't the case. It's actually a primary pathogen. So what we see now in our sea lions, like all the time, this has now become a fairly regular endemic disease, is they'll often just present it as a sudden death. And what happens is they get abscesses in the lung. This is the lung here and the chest cavity. And these abscesses will get quite large and they'll rupture, and then they'll release the bacteria into the chest cavity, and you get a chest cavity just filling up with pus. We call that pyothorax, and the lungs get compressed and they'll die from sepsis really quickly. Like they'll often be rescued just with some minor respiratory difficulty, and then they'll just drop dead in transit. Now, interestingly, over the last 3 years or so, we've seen a lot more of this. So here is a graph showing from 2017 to 23, and 2022 was actually the highest year we had for this purple line. But you can see 2023 is tracking right very close to that. So we're right on track with being another really bad year for it. And then 2021 was also a bad year for it. So exactly what's going on out there, we don't know. So Klebsiella pneumonia, or KP, it's a very common cause of pneumonia in people. And before antibiotics, as I said, it was a major killer. Now, this particular strain of it, called hypermucoviscus, or HMV, was first discovered in the late 1980s, early 1990s in East Asia as a hospital-acquired infection. And then from there, it spread globally. And it was, you know, obviously not long after that, like 10 years or so before it got into sea lions, both in New Zealand and the sub-Antarctic and in California. Now, how that ever happened, I don't know, but most likely from people, somehow from people. And our surveillance work in New Zealand showed that we definitely know it wasn't in the population before 2002. Less so we know about the California situation. And the isolates that we made in New Zealand, when we tested those genetically, they were clonal, so they were all likely from a single source of infection. So most likely it came from people. Nobody lives on the Auckland Islands where these sea lions are, but there are researchers there. There's a fishing fleet, squid fishing fleet that goes there. There's ecotourists, you know, there's any number of people who could possibly have brought it there. And our current research with our collaborator Esteban Soto at Davis is looking at the genetic diversity of these recent isolates from California. And the goal of that is to try and find out whether, you know, what's happening now is all because of one particular genotype or something. So he has a star student, Madeline Thompson, working on that. And this is a poster that she just recently presented at a conference, looking at the genetic diversity in the isolates we've made from our TMC animals. So they're actually a little bit more diverse than we were thinking. It's not just one particular genetic strain that suddenly appeared and taken off as the main culprit. Sarcocystis. Now, sarcocystis is it's a protozoal disease, and it's interesting because we've known about sarcocystis as a pathogen for quite a long time. In sea otters and harbor seals, it's known to cause encephalitis, same as toxoplasma does. Toxoplasma you might be more familiar with because it's the protozoan from cats, and it spread in cat poo and then gets into other things, including people, and can cause encephalitis and blindness. Sarcocystis is different though. It's a cousin, but its host is the Virginia possum, which is not native on the West Coast. It comes from the eastern woodlands. It was brought here, bringing its parasite with it. Now, this parasite behaves in the same way as toxo. It gets shed in the poo, gets into the water, gets into the fresh water, into the ocean, and it's quite resistant. Now, the presentation is different in sea lions. What we found in them is that they develop this muscle disease or myositis, and they'll often present with confusing clinical signs because it depends on what muscles are affected, what signs are shown. So if it affects the muscles of respiration, like diaphragm, the chest muscles, the larynx, they present like they have pneumonia, but their lungs are actually fine. It might just affect the striated muscle of the esophagus, and they present with megaesophagus, not able to swallow, and often being just wasted from that. Or it can affect multiple muscles in the body, and the whole body will appear wasted. Now, we had one case of this back in 2010, an adult female sea lion who presented with that muscle syndrome. And back then they did a biopsy, showed that she had muscle inflammation and that sarcocystis was present in it. So they treated her with an anti-sarcocyst or an anti-protozoal drug. She seemed to do okay. She was released. And that was the end of the story. Nobody knew anything about how she did or anything. And then we didn't see it again until 2016. And since then, we've seen a lot of cases. And this is how it presents. So they'll often look really, really emaciated, like really sunken muscles, no body fat either. And when you take off the skin, you can see all these muscles are really pale and streaky. And you can just see how wasted they are, like really, really thin. And this is the larynx, and this is the, so the epiglottis is over there, and this is the opening into the airway. And the muscles here on top of the larynx should be big and bulky. And these are the muscles that open the larynx to allow breathing to happen because they're active breathers. They don't, the default position for them is the larynx to be closed. So they need these muscles to actively open the larynx to breathe. And if these muscles are all wasted like this, then they have a really hard time breathing, and you can hear them really harsh lung or breathing sounds trying to do that. The diaphragm can be severely affected. You shouldn't be able to see through a diaphragm like this. So this is the diaphragm taken out and just held up to the light, and it's paper thin. It should not be like that. And these are other muscles of respiration. This is in the body with the chest wall. See how streaky they are. They should not have these white streaks. This is all necrotic or dead muscle fibers. And this is one with megaesophagus. So the outline I've shown there is this big distended esophagus. There's the lungs and the heart's down here. There's the aorta. But that esophagus should not be distended like that. And that's completely blocked and plugged with food. So she's not able, and regurgitate. So she's not able to get that either up or down, and is not able to eat. And again, she's got the streaky, pale esophagus, diaphragm. And if you look at that under the microscope, so this is a diaphragm here from another animal. It's really thin and very blue. And the blue is dead or necrotic muscle cells, some of them all mineralized like here. And then in between where there are a few normal fibers, you can see these blue cigar-shaped things. These are the sarcocysts. And at higher power, you can see one here. It's got its little capsule around it and all these little tiny baby sarcocysts inside, all these little zooids. But all of this muscle, either side of it, is all dead. All this blue stipply muscle is dead. And then you still have some live cells, but these are either leukocytes, like lymphocytes and macrophages, or satellite cells, which are trying to regenerate the muscle. But effectively, all of the muscle, either side of this one live fiber that has the cyst in it, are dead now. So it's very nasty. And another example here, this is showing the polyphasic nature of it. So you've got myocytes that are dying, or the pink ones, and then you have others that are regenerating, or these blue ones with all the nuclei lined up in it. So they can recover from it if they get, nursing and stuff, but usually it's a one-way. And if you want to confirm that the sarcocysts are present, you can do special staining for them. So here's the cyst, and all of this browny stuff is a special immunostain showing that it's positive for sarcocysts, and all these little dip dots here, these are all the tiny little zooids actively invading other muscle cells. Okay, so the life cycle, as I said, the possum is the main host for it. It's passed out in the poo. The sexual stage of it occurs in there, and then in the poo you pass out the sporocysts. They get taken up by other animals. In this case, it's showing a horse, because this is a major disease in horses, and it's called equine protozoal myelitis, and it's a major cause of neurologic damage to the spinal cord in a horse, and paralysis. But it can get into a whole range of other species and cause either encephalitis, as I mentioned, or this muscle disease, as we see in the sea lions. And this is an example of land to sea disease transmission. So toxoplasma is the other main example of that, and it's a huge disease in sea otters and harbor seals. Okay, so since 2016, when this appeared again after that, first one in 2010, we've seen a steady number of them every year. And that was 2022, 23. We've had another bunch of cases, so it's something we see now every year. And we published a pathology paper on it, so describing all that pathology. We also published this paper on the clinical signs, and I mentioned how it varies depending on what muscles are involved and how it's diagnosed. So that we had a hypothesis when it appeared in 2016 that perhaps this was a completely new strain of Sarcocystis neurona, and not the one that's generally circulating in the possums here. So a reason for that was that this particular strain here, genotype 13, was found in the Pacific Northwest region, and that was a new genetic strain of it. So we thought, okay, we'll do some sequencing on ours and see if it's like that. However, turned out it wasn't. What we have in our sea lions are the types that are already known to be widespread in North America, type 2 and type 6 strains. So it's not that it's a new strain. What we're seeing is a different response. So the sea lions are responding to it in a way that's different from what other animals do, and it's acting more like it's an immune-mediated disease. So somehow the parasite is triggering an immune response against the muscle cells in the sea lion, and it's a specific sea lion response to the pathogen. Okay, so why did it suddenly seem to increase in prevalence in 2016? Well, we know from the work that's been done over 20 years with sea otters for both toxo and sarcocystis is that it's associated with runoff from land. And they in sea otters, we tend to see a spike in infection in anywhere between like February, March, and April, when you get all of that, runoff from the winter rain coming into the ocean at that time. And that's when we see a spike in cases in sea otters. So it's quite possibly related to that. And then and because of that, then related to climate. And it's interesting how this increase in prevalence occurred or seemed to occur starting in 2016, around the same time we were having these thermal anomaly events happening in the North Pacific. So are they related? Are they not? We don't really know, but it's quite possible that changes in rainfall or runoff might have happened there, or else changes in prey distribution on what the sea lions were feeding on could have had an effect as well. So a recent project, again, with a UCD STAR student that was just presented just very recently, was developing a new lab test to look at the seroprevalence of this in sea lions, so we can get a better handle on the disease ecology of it and whether there are any hotspots associated with it. Okay, so getting on to the toxin stuff. So domoic acid intoxication. Let me know if I'm going too fast or too long. It's already 7:15. So am I good still, or how are things?
Everyone is still here listening. We're great. Just take your time.
Okay, I've
yeah.
just got two more topics to cover, and it's toxins and then the great whale mortality. So we're nearly done.
Nice.
Okay, so
Don't worry.
okay. Domoic acid poisoning. And I'm sure everybody there probably heard of this because it's been huge in the news over the last 2 months or so, with the big outbreaks that happened in Southern California that killed hundreds of dolphins and sea lions. And it's funny when you hear people calling it demonic acid instead of domoic acid. Anyway, so this is the outbreak that happened in Southern California. And as I said, hundreds and hundreds of sea lions were affected by it, and also common dolphins. And a weird thing about this recent bloom down there was that it happened in deeper waters offshore, and that's how so many dolphins were involved, apparently. Luckily, it didn't come up into our TMMC range very much. We had maybe a dozen or so cases from San Luis Obispo area, but we haven't had anything like they had in Southern California. Okay, so it was first diagnosed as a disease of California sea lions in 1998, and that was from an outbreak that were something like 70 sea lions with seizures turned up at TMMC in Sausalito in one weekend. I heard the story, and at the time they were thinking it was morbidity virus, that distemper virus, because of the neurologic signs. Nobody suspected it was a toxin. So they did all the testing for viruses and nothing showed up, but luckily they had collected enough tissues and samples to test for toxins too, and that's when they discovered it was domoic acid poisoning. Now, domoic acid was first discovered back just over 30 years ago in Canada, because it caused an outbreak of shellfish poisoning in Montreal, and the shellfish had come from Prince Edward Island, the Prince Edward Island mussels. And I think, I don't know, 7 or 8 people or something died in that, and a whole bunch of other people were sick. And that's when it was first discovered. We now know it's all around the Northern Hemisphere, but it's definitely very prevalent here. But 98 was the first time it was recognized as causing an issue for not just sea lions, but pelicans and anything that eats anchovies, essentially. And there were probably 2 earlier outbreaks in 91 and 92 where there were animals reported with neurological signs, but it was never diagnosed back then, but it's quite likely that's what it was. And then so for the last, 2 decades, the 90s and the 2000s, these events were kind of fairly sporadic and spread out, some severe, some not. But since over the last 10 years or so, these have become increasingly frequent and extended both geographically and temporally. And in 2015, if you recall, there was a domoic acid bloom that extended from Monterey all the way to Alaska, and that counted for killing whales as well as the smaller marine mammals. So it's hard not to think that this is also related somehow to these climate changes and thermal anomalies that are happening in the North Pacific. And over the last, since it was discovered in the late 1990s, TMMC has kind of been at the forefront of doing research on this, and a ton of papers have come out, mostly sea lion work, but also sea otters and other things too. And as I said, we've cases every year, but if I just broke this down by those early years, 98 to 2009, where these events were kind of spread out and sporadic. But since 2014, we've had them practically every year and almost in any season. It's not just limited to the late summer or early fall anymore. And showing, some of our data from sea lion emissions at the center. So these peaks here in recent years have coincided when those two thermal anomalies happened. And the cause of it is a toxin produced by a diatom called Pseudonychia australis. It's a neurotransmitter agonist, so it acts like a neurotransmitter, and it binds onto glutamate receptors in the brain and the heart. Now, these receptors, when they're triggered by something binding to them, are excitatory, so they will excite the cell that has the receptor and cause it to fire off, and it will do so repeatedly because this toxin is bound to it. And that will just use up the ATP or the energy source in the cell, and eventually the cell, the neuron or whatever it is, will die from exhaustion, and that's what happens. So the parts of the brain that are particularly dense in these receptors and heavily affected are the limbic system, and that includes the olfactory system, or tracts, the amygdala, the hippocampus, and the parahippocampus. And those areas are associated with things like memory and spatial memory, geographical recognition, all that kind of stuff, and emotion. So, and then in the heart, the part that's affected is the very important conduction system. So animals that have that affected will end up with cardiomyopathy. Now, I mean, it's bad for the animal, but it's actually good for the pathologist, because this gives you very specific places to look and very definite lesions to look for. So in the hippocampus, the hippocampus has this curlicue of neurons. So this part is called Ammons horn, and this part here is the dentate gyrus, and those two areas are affected a lot. And you also get the brain tissue in between affected. So this stratum lacunum moleculare is affected. So all these little holes are vacuoles. And if you look closely at this, so this is a low power view, but if you look at that at a high power view, you can see some normal neurons like that one, nice, plump, nice nucleus. Then you look at these ones that are marked with the yellow. You can see they're small, shrunken, very dark nucleus. These are all dead neurons. So these ones have been overexcited and they've burnt out and died. And once that happens, they're gone. So they're just removed. So these ones are already fading away. And then you're left with these holes in the brain. So these neurons that die are never replaced. And then in response to this death, you get inflammatory cells called glial cells coming in to try and remove them. But yeah, so it's sort of a one-way street. You lose your neurons, that's it. But for any one event, quite often, not all of the neurons will die. So the animals often survive a single shot of toxin. And then you get into a situation where you've got chronic DA intoxication. So you'll have loss of tissue from parts of the brain. And they may survive with that for some time or have just periodic or random seizures. So this is an MRI scan of a Guadalupe fur seal who was having these sort of random seizures, and the MRI scan shows that on this side here, which her left side is on the right of this, there's loss of tissue here from the hippocampus compared to on the right side, which is more normal. Now that animal, once it was diagnosed, she had that, she was euthanized. And then this is a slice through that of the actual brain that's been fixed in formalin. And what you're looking at there is the thalamus, the corpus callosum, cerebral hemisphere. And this bit that curls in there is the hippocampus. And you can see the right side and the left side. Now, the right side was the side that was more normal. So you can see it curling around here, parahippocampal gyrus is here, the Ammon's horn is there, the dentate gyrus is there. So all the parts are present on the right side. On the left side, it's very shrunken, and you've got these two big holes, but that's actually an expansion of the lateral ventricle. So the tissue of the hippocampus is gone, it's atrophied, and you've got expansion of the ventricle around it. So that's a chronic DA case. And then you get effects on the heart. This is a sea otter that died from cardiomyopathy, and so she's in heart failure, and fluid is now built up in her abdomen and her chest. And you're probably familiar with people that are congestive heart failure. They have to have diuretics to get rid of the fluid out of their body. So this is what happens if you don't have diuretics to get rid of the fluid. You just fill up with fluid. And the heart goes from being like normal looking to that to looking like this or this. And if you make a histological section of the wall of the heart, the pink tissue here is our normal muscle cells in the heart. The blue is all fibrous tissue that's taken over where myocardial cells are being killed and replaced by fibrous tissue. So a heart like this is not going to function very well. Okay, so we've been doing, like I said, a lot of different research projects over the last 2 decades on domoic acid poisoning. This is just a recent one that this student got Josh just defended as a PhD a few weeks ago. So they were looking at the expression of proteins in animals that had that were normal, animals that had acute DA poisoning, animals that had chronic, and animals that had acute infection, acute toxicosis on top of an old chronic lesion. So my role in this was to come up with these cases, the acute, the chronics, and the acute on chronic. So it meant looking at a lot of histology. So here's, again, normal hippocampus, left side, right side. And this one is, nice and symmetric on both sides. There's your nice curly Q. Hippocampus is derived from the Greek for seahorse. So it has this little seahorse appearance to it. And so you can see on both sides, it's symmetric and it's nice and plump. And there's the ventricle around it. So that's how it should look. And there's Ammon's horn. CA means cornu ammonis. 1, 2, 3, and 4. Those are the 4 segments of it. And then the dentate gyrus is here. So they're all the normal parts. So with acute toxicosis, you have to look at high power to see something happening, because at low power it looks okay. But at high power, you can see all these dead neurons, all these really pink neurons are dead. So that's an acute case. This one is a mild chronic atrophy. So it's shrunken a bit. There's more expansion of the ventricle, particularly on the left side, more so than the right side. So that's a mild case. A more marked case, it's asymmetric. Left is worse than right in this one. And then you see much greater expansion of the ventricle and shrinking of the hippocampus, and a lot of vacuolar change in it too, and loss of neurons from the Ammon's horn and the dentate gyrus. And then as you get more severe, again, severely shrunken now, and loss of definition of the Ammon's horn and the dentate, they're practically gone on the left side, more so than the right. And this is one where you've got a chronic loss of the tissue, and then superimposed on that, when you look at high power, you see this acute loss of neurons from a more recent intoxication event. So that was that study. Now, this is probably the one of most interest to you guys because it is started with Kringles. Kringles stranded up at Fort Bragg 3 years ago, maybe now, I think. Subadult male. He was kind of skinny condition. Now, this guy had a number of things wrong with him. He had leptospirosis for sure, and that was actually the main reason why he stranded and why he ended up dying. But when they did a routine X-ray on him to check out his lungs when he was admitted, they saw that on the X-ray there was this sort of bony proliferation around the humerus. So this is the shoulder joint, that's the humerus, that's the elbow joint, and the radius and the ulna. So you've got this fuzziness here around the shaft of the humerus, which shouldn't be there. So they saw that and they thought, oh, that's kind of weird. So then the X-ray the other flipper and it had the same thing. And he eventually died of the leptospirosis anyway, and then came to necropsy. And when we got him, we knew about this bone change, so we went looking for it, and we confirmed that he had this. So we took the bones out, cleaned them, and this is what they look like. So Kringles is on the left there, and a control age-matched male, subadult male is on the right. So you can see the shaft of the humerus here should be nice and smooth. And here on Kringles, it's got all of this bony proliferation all along it, quite marked and chronic. But the actual joints are spared. It's really just on the shaft of the bone. So that's the humerus. Same thing here. This is just another angle on the humerus of Kringles. And then here's the short little femur. So that's the head of the femur, the ball and socket joint of the femur. And again, the shaft of the femur is all thickened by this proliferation. And this was a cross section of the bone before the other one before it hadn't been cleaned. So this was just fresh bone. And you can see this is all this bony proliferation on the outside. This is the marrow cavity on the inside. Now, what you can see here, this dense bone, that's the normal depth of what the cortex should be. And everything out here is all new bone proliferation. This one shows it even better on this femur. So here's the normal cortex, the bone marrow in the middle, and then all of this here, like 3 times as much, is all new bone proliferation outside of the cortex. This is a figure from the paper we wrote on that. So there's the femur, whole section, cross section. We also have bone. We have changes in the teeth. We also have changes on ribs from another case. This is one that Sarah found where she only found some of the bones, but they had this bony change. And then this is just more histology of what's going on there in the bones. You have your normal cortex, and then you have all this bony proliferation happening. So this is a mandible from a sea lion age control one, where it's just normal, just flesh cleaned off it, no bony proliferation. Here is one of the other animals with the fluorosis, with all this bony proliferation here, all this honeycomb of bone on the outside. This is from the os penis, again, all this bony proliferation, and then just again histology showing those changes at the microscopic level. Okay, so when we first saw this, we came up with a differential list as to what causes it. So from what we know with domestic animals and wildlife, is that in domestic animals anyway, most of the cases we know about are genetic and associated with particular breeds of pigs or dogs. Most of them are in dogs. And then there's also this disease in people, Caffey's disease in children, which is, again, has a genetic component to it. Then there's a couple of inflammatory diseases that can cause it. One is called hypertrophic osteopathy, or Mary's disease. Now, this is where there is something in the chest cavity. It could be a lung cancer, or it could be an inflammatory disease like pneumonia, or it could be anything in the chest cavity, somehow triggers this kind of proliferation to happen in the long bones, particularly of the forelimbs, but it can be elsewhere in the body too. Now, it's not that common, but I've seen a few cases of it in dogs. And then there's this protozoan, Hepatozoon americanum, in dogs, which tends to occur more in Eastern North America than here. So we weren't really thinking that. We didn't see anything in the chest that could explain that one. So that then left us with these two. So fluoride poisoning or vitamin A poisoning. So first thing we did was we sent some bone off to UC Davis to check it for fluoride. And sure enough, came back screaming high levels. So this is Kringle's. So this is his humerus, his femur, and rib. And so levels in the thousands parts per million dry weight of his bone. And then by comparison, if you look at like a control cow, it should be, less than 1,000, or a cow with fluorosis anywhere up to 3,000. But this is like 3 times that level. So really, really high levels. So then that begged the question, well, do marine mammals have higher levels of fluoride anyway? So we got some sea lion controls. So again, age and sex match controls, and looked at them who didn't have the bony lesions, and their levels are higher than in the cattle, but substantially lower than in Kringle's who had the disease. And then we expanded that with a few more. So we looked at more bones from Kringle's, and we looked at bones from the other 2 cases that came from Fort Bragg area, and 3 control age matched controls. And, significantly, that all of the cases had much higher levels than either the sea lion controls or in terrestrial animals. Okay, so fluorosis, as I said, has been described in terrestrial animals, and I was familiar with it from Australia because in Victoria, where I was at University of Melbourne, we would see cases in macropods, so kangaroos and wallabies, from a particular area in Victoria that was downwind from an aluminum smelter. And they had all of these changes: hyperostosis, the osteosclerosis, osteoporosis, all these various bone changes. They also had changes in the joints, which we didn't see in the sea lions, and they had a lot of changes in the teeth, but then they were exposed to it right through their life. So from, the mothers would have it, their offspring would have it, and because the developing teeth would be exposed to the fluoride, the teeth would develop all sorts of cavities and weakness that we didn't quite see so much of in the sea lions. Okay, so what are we doing now about it? Well, luckily, when we sent these tissues off to UC Davis, Robert Papenga, who is the toxicologist at the lab, he just started a new toxicology resident, Chelsea Sykes, and she was looking for a research project. So this became her research project. So we started collecting a lot more bone from a whole range of sea lions, different age classes, and from different areas, and also collaborating with California Academy to get samples from their collection, and more cases from you guys' area, and looking at this to look at age classes and all that, how that might play out as to how prevalent this is. And we had hoped that we'd also include in this looking at prey species, so looking at fish and all the various things that sea lions eat from Northern California and Southern California, to see if we could see any difference in that. But that transpired to be a much bigger project than we had hoped to get into, because turns out sea lions have a very, very eclectic diet, and they have, at least 24 different common things in their diet. So to test all of those in enough numbers to answer this question would be a lot of work. So right now we're limited just to working out who in the sea lion population has this and how prevalent it is. Okay, so down to the last thing. So the gray whale unusual mortality event, or UME. Okay, so gray whale mortalities is something we see every year. We see maybe half a dozen in a year. That's kind of like typical for our length of coastline. But during the UMEs, we see a lot more. And there have been two UMEs, one before my time, which was 1999 to 2000. And then this more recent one was started 20 years later, 2019, and has gone through to it. It's not officially closed yet, but it effectively is done by now. So in both of those periods, the first one and the second one, there was significantly more dead gray whales than the average. And the other thing is that calf mortality, or not calf mortality, but the number of calves born, so calf recruitment into the population is something that varies a lot. It fluctuates here, like these bars you can see here. And sometimes that coincides with the UME event, as it did in 1999-2000, and here again in 2019-20. But there are other times when calf production also decreased, like it did at that point there, 2008, 9, 10, but there was no associated adult mortality. So the calf production is probably something that varies with the fitness of the adult females to be able to carry a pregnancy, and maybe related to environmental factors. But it also seems now that the adult mortality is, and I'll show you why. That's just those two events. Okay, so over this period of this last UME, there was 590 dead gray whales recorded between Canada, Mexico, and the US. So that's like from Alaska all the way down to Mexico. And over that period of time, so from the last census, which was in 2016, to a census in 2020, 24% of the whales had disappeared from the population. Pretty significant amount. And the same had happened back in 1999-2000. It was about a quarter of the population died. And the this graph here just shows you 18-year average in the gray, and then what happened in those UME years. So 2019, it was the worst of this last UME, and then followed by 2020, and 2021, 22 is dropping right off. So what we saw is that a lot of these whales were just in very poor body condition and often just covered in lice. Like all of this pink stuff you're seeing here, these are cyamids or whale lice. Now, they're not a pathogen as such. I mean, all whales have them, and they will eat dead skin and stuff like that, but they don't, they're not really a parasite. But if the whale is debilitated and not swimming properly, not doing its normal grooming and as they do, rubbing on the bottom, etc., the numbers will build up enormously in a debilitated whale. And what we saw in a lot of these animals, which is very poor body conditions, here's another example of one here, subadult male. And what we did to determine that was not just measuring the depth of the blubber, but actually looking at the composition of it histologically. So this is histology of the blubber from the outer part of the blubber layer, the middle, and the deep. So the more open the tissue is like this, these are the adipocytes, or fat cells, and the pink is just the structural tissue of the blubber, like the fibrous tissue, connective tissue. Now, in a good conditioned animal, there should be nice, big, fat, plump fat cells in here. These ones are pretty shrunken. And these are even more shrunken. The more blue there is, the less fat there is. And then you get down here to the deeper blubber, and there's no fat here left at all. So we did this for every single whale. We looked at how much fat there was in the blubber. So we didn't find any single cause of death in these hundreds of animals. We had roughly 1/3 of them were in our area anyway, were ship strike animals. Malnutrition was a common finding. Some had evidence of killer whale attacks, or there were combinations of these. And for a lot of animals, not so much in our area, because we tended to get to them pretty quickly, but in other areas where there was delay in getting to the animals, often decomposition was a major factor, and then they just couldn't find out why it died. But for all of the animals where testing was done, no pathogenic organisms were found. So no significant bacteria or viruses or protozoa or fungi or anything like that. Biotoxins were tested for, and some had some had levels of saxitoxin, some had levels of domoic acid, and for many it was not detectable. And even where levels of those toxins were thought to be high or moderate, we have no way of knowing how significant that is for a whale, because we have no pathology like we have with the sea lion to go along with that to say, yes, it's doing something. But the main common factor for all of these years was the poor body condition. And that was corroborated by other field studies. So the drone work and photogrammetry that's been done on the migration route, between Canada and Mexico, and the work done in Mexico while they're down on the calving grounds, all of that confirmed that whales were in poor body condition from when they left Alaska, when they should be in good body condition after spending the summer feeding, down to Mexico, where they don't do a lot of feeding or any feeding, and then the migration back up. So all along that migration route where these animals were photographed and measurements taken, they were in poor body condition. So what do we know about this period? Well, in general, we know that the sea ice covering the Bering Sea and Chukchi, where these animals feed, has been receding at an alarming rate. And then in conjunction with that, the benthic prey up there, like the amphipods that form the main part of the gray whale diet, have been shifting their distribution north from the Chukchi into the Bering and further north. So whales who have traditionally gone to a particular area to feed are now in areas where there's no or little food. Those that are would historically have gone further north probably still are getting fed, but large segments of the population are not getting the feed unless they move and shift their feeding grounds. So we do know that gray whale distribution has changed based on some published work. So they are shifting in response to the lack of prey in their traditional areas. We also know that both the earlier UME and then this recent one were preceded by unusual events. So a strong El Nino for the first one, and then these North Pacific thermal anomalies for the recent one. So yeah, lots of changes in where the whales should be feeding and getting their groceries. There's a lot of changes happening up there that are affecting them. But the underlying question is still not really been answered. So whether is this climate change and change in the Arctic, is this directly affecting what they're feeding on? And so, the amount of groceries they got access to, or is it something to do with the number of whales? Are they outstripping their resources, so getting to carrying capacity of the environment? And prior to both of those UMEs, the most recent whale censuses before that had shown that the population was around 26,000, 27,000, and then it crashes and they lose a quarter of the population and it slowly builds up again. So it's quite likely that we are in some kind of a multi-decadal, like a 20-year cycle of this happening, and this may be the norm for the population now. And who knows what effect climate change on top of that will have, with the change in where their prey is moving in the Arctic. So still lots more research questions on this. And that is it. So sorry for taking up so much of your time. And if you have any questions, I'll be happy to answer them.
No, thank you for your time, Greg. That I know you're a busy human, but I see that most everybody stayed with us and thanked you and said, this is good stuff. Thank you for such a thorough discussion. In your Canada days, did you ever collaborate with Dr. Joe Gerasi or Guelph? Just curious. I don't know if I said.
Yeah, he was my PhD supervisor.
There we go.
Yeah.
Nice. And
so I was his last PhD student. In fact, Joe had actually left the Ontario Veterinary College to move to Connecticut to be the research director at Mystic before I'd even finished my PhD. So I turned the lights out in the lab, but was the last one out of there. But yeah, he was my supervisor.
Nice. Small world when it gets to that high level.
Yeah. Yeah, and that's what I said at the beginning, you know, when I was, you know, finishing up my vet degree in Dublin and looking for somewhere to go to do marine mammal graduate work, there was almost nowhere you could go. Joe at Ontario Veterinary College was the only professor doing this kind of work that I knew of, and I think that there was. I mean, there were other people doing marine mammal research in different places, but often just like associated with aquariums or to the Navy or something. Nobody in an academic institution.
Right. Well, I'm glad that it's turned this direction to where these things are really getting studied. Just seems so very, very important. So with that, kudos and appreciation to you, and take good care.
Yes, thank you so much. That was great.
Lovely to meet you all, and hopefully we'll do it again sometime.
Right.
Stay in touch. All right. Thank you.
Bye.